Welcome back to Chapter 13, Part 2. So we had been discussing Darwin's history before he wrote Origin of Species. And he saw all these different versions of species and these different traits. And he was greatly influenced by Lyell, that geologist. Because they had thought the world was so young, evolution wasn't really seen as a possibility. But given the fact that the Earth is really old and has taken a long time to be shaped, that gave Darwin time for his ideas, so that you could have these millions of years for these slow changes to occur. Darwin called the ideas in The Origin of Species descent with modification. There, he said that organisms today descended from ancestral species. So ancestors gave rise to what we have today. And that natural selection was the mechanism for descent with modification. How over generation you saw changes. So looking at a phylogenetic tree showing the relatedness of elephants, if you look back, there was a common ancestor more than 34 million years ago, and that gave rise to the different species of elephants. Those that are still extant or alive today, the Asian elephant, the savanna elephant, and the forest elephant. In this area, we've got all different species that look somewhat similar because they come from the same ancestor long ago, but over time you've seen different changes. Why would they say they're related? What evidence do they have? It's not just based on looks. Let's look at the evidence. Big one is the fossil record, biogeography, comparative anatomy, embryology, molecular biology. Let's look at each of those. So first, fossils. So those are the remnants or impressions left by organisms that live in the past. You'll generally see this in areas where there were water. That's the best place to make fossils. And over time, you'll have fossils that get stuck in there. Some organisms make for better fossils than others. You'll see things like plants in there and bones much more easily than you will any soft tissues. Those are generally degraded before a fossil would set in. Paleontologists are those scientists that study fossils. So they look at the layers and what they have inside them. So they can see like those transitional species where you've got like whales with hind limbs, for example. Another would be like a fish with legs. There's a series on Netflix called Your Inner Fish, and it's 3 episodes, and it's done by a paleontologist slash comparative anatomist, Neil Shubin. And he was looking for a transitional fossil between fish and moving on land. And so he wanted to look for the like reptilian head shape that's more flattened rather than long, and 4 limbs. And so he knew kind of what strata to look in. The Devonian is this time of fishes, so he had to be kind of late that period. And after a few years of searching, he finally found it, and they named it Tiktaalik. Tiktaalik was the link between fish and reptiles. Found it in northern Canada, and it was the local Inuits' word for freshwater fish. So that's the mold of the bones that they found, as well as the representation of what they would think that individual looked like. And here's Archaeopteryx, basically a reptile with wings. If you didn't know, look at your chickens that you have, if you have any. They run around like little reptiles. They're little dinosaurs. Next is biogeography. So that's the geographic distribution of species. So before we had these plates shifting the continents around, there was one large continent called Pangaea. And you could see through biogeography how if you put these together, the ranges of the animals make a lot more sense of where you're finding their fossils. And that showed Darwin that when you look at the parts of South America and Africa that have very similar ancestors, maybe they evolved from a common ancestor. One problem other individuals had with Darwin's idea is that they thought the animals that were in their environments were perfectly designed to be there. But that's not true. You've got different types of animals in similar habitats. Australian desert animals look very different than the United States animals. And then there are similar animals in different habitats. The explanation is that the animals in similar areas had common ancestors, whereas those in similar habitats in different areas don't have as common of ancestors. Australia is a really interesting case because they have marsupials. The only marsupial we have here is the possum. Mammals in other areas they think outcompeted marsupials. Without that mammal presence, marsupials were able to grow and survive and produce offspring more easily in Australia. Another area that I'm very interested in is comparative anatomy. So that's looking at body structures between different species. For example, looking at our bone setup or our muscle setups that are the same in dogs and people. They have different slightly structures, but for example, with arm bones, we've got our digits, our wrist bones, our short bones, two bones, and a long bone. And we learn a lot about people from studying other animals. A lot that I teach in the anatomy textbook is based on what we've learned in other animals. So comparative anatomy looks for homology. That's similar structures due to common ancestry. So this was what I was talking about. You've got the phalanges, the finger bones, the wrist bones that are short, two bones together, and one long bone. And you see that in all these different animals. And that's because we have common ancestors. Homology, homo, one, they come from one ancestor. You can also see this in embryology. So embryology is the study of structures that occur during development. What's really cool is in vertebrates you've got, most of those are chordates. Chordates have similar things in common. They've got a postanal tail. They have gill pouches. They have a dorsal hollow notochord. What does that mean? We have gill pouches during development. Those become part of our inner ear bones in the jaw that I was discussing before that we had in common with reptiles. We've got a tail. We can learn a lot by looking at, for example, shark embryos to understand our embryos. And here's an example of different embryos from different animals. And you can see there are very similar structures. This supports the idea of evolution. The newest way to study evolution is molecular biology. And that's looking at genes. So our DNA. Animals, plants with more recent common ancestors are going to have more DNA in common. So we've changed around the phylogenetic tree how things are related based on our genes and the different versions of the genes. So here you've got 5 species with the same gene. They share a common ancestor. And the one with the green share a common ancestor, and that makes sense based on their genetics. Some parts of our genes are going to be highly conserved. They're very important, like the functions for making an eye or for a hand. They're going to be highly conserved through the species, so any changes we'll be able to trace. And what we look for the simplest explanation possible, where you'd have to have the least amount of mutations to make these fit together. That's called parsimony. The easiest solution is probably correct. Looking at the molecular biology of monkeys, you can see how similar the genetics are between different species. So Old World monkeys are the least similar. They branched off from a common ancestor the earliest. Then gibbons, orangutan, gorilla, humans, and chimps. I think this takes millions and millions of years. Now we're going to talk about adaptive evolution and natural selection, how we select for these different traits. Darwin looked at finches on the Galapagos and their beak shape. Based on a bird, the finch's diet, their beak shapes can be different. So like a large beak would be good for opening nuts, whereas smaller beaks might be better for eating fruit, or like dexterous beaks for getting into areas. Let's say there's a really dry year and there's only seeds. The birds with the bigger beaks that can eat the biggest seeds are going to survive the best, have the most offspring. So in the next generation, you'll see bigger beaks. And there's actually a couple that lived in the Galapagos. They wrote a book called The Beak of the Finch, and they did this. They studied, they measured the beaks of finches over different seasons, knowing how dry it was, how wet it was, and they looked at those changes, that adaptive evolution that took place over time. So let's look more at the theory of natural selection. The two observations he made. One, overproduction. You're going to send out more individual offspring than the habitat the environment can support. This leads to a struggle. Some individuals are going to be better at surviving, not getting eaten, thriving in these environments. The second observation, there's individual variation. So individuals of the same species can look somewhat different. There's variation, and that variation can be heritable. It can be passed on from the parent to the offspring. So if they have some sort of successful trait, that can be passed on to their offspring. Therefore, if you have too many individuals and you have variation in them, there's going to be a differential reproductive success. That's natural selection. Some individuals are going to pass on more genetics, are going to have better offspring, and those offspring are going to survive and create more offspring. So for example, we already looked at the pesticide-resistant in insects. Those that were resistant survived better. They passed on more genetics. And same thing with antibiotic-resistant bacteria that we looked at the beginning as well. You've also got, for example, viruses. Viruses can have natural selection. HIV mutates quickly and can often mutate to be drug-resistant. That's why we're always looking for new ways to treat HIV. And it's important to realize that this is survival of the fittest. And what we mean by fittest is that they have the most offspring, they're the most successful. It's not that they're the strongest necessarily, but they've passed on their genetics to the most offspring. Now let's look at the modern synthesis. So this is putting together what Darwin found with genetics. He knew that they were passed on, but he didn't know how. He didn't know the rates or the traits that Mendel was working on. Unfortunately, Darwin hadn't ever heard of Mendel or what he was working on, even though they were alive during the same time. They didn't see how these fit together. We've already discussed that individuals don't evolve. They don't change their traits. That happens at the population level. So what is a population? It's a group of individuals of the same species living in the same area at the same time. So in a specific forest, you've got the deer that live there. That's a population of deer. It's the smallest unit that can evolve. So population genetics looks at the genetics of the population. It looks at the rates at which you see different traits. So how many beetles are yellow versus brown in that population, for example. And then it looks at how those changes over time. And then you see that over time you might have more yellow genetics in there for yellow beetles than brown, and we would call that evolution. Now some people, they have a problem with this because genes are carried in individuals, not over populations. But that's where we see the changes. We see it at the population level. We see how overall you have more yellow beetles than brown. Then individual variation is present in the populations. You can have the different colored beetles. And not all of it is heritable. Some of it can be due to environment. But then that's not what we would look at for evolution, natural selection. We would only look at the genetics. What can be passed on? That's the important part of natural selection.